In this video, we will discuss about the after electron transport chain, the Calvin cycle in the dark reaction in the stroma of the chloroplast will be occur. In this way, here is the ferredoxine will produce the ferredoxine NADP plus reductase enzyme used to produce NADPH, while the ATP synthetase enzyme when move the proton out from the lumen into the stroma. In this way, the 4 proton will be moved out to produce 1 ATP, and this ATP and NADPH is the energy producing step during the light reaction lead to dark reaction in the stroma. For example, Calvin cycle. That's Calvin cycle required light, not light reaction, but dark reaction. Dark mean it is independent, light independent reaction. For example, this is the Calvin cycle, and it is let's begin to understand with concept. So this is the light will be fall on the tree. For example, green photosynthetic organism autotrophs. This is the photosynthesis, but the carbon dioxide fixation, the 3 molecule of carbon dioxide fixation, which that will not require light, but requirement of the light on the step of electron transport chain to produce ATP and NADPH. So let's begin to understand. Here is the 3 carbon dioxide will be fixed into the ribisco in the presence of ribisco enzyme, which that is present in the chloroplast in the stroma. So stroma contain ribisco enzyme. So this is the ribulose 1,5 bisphosphate 3 molecule RUBP will be fixed, and in this way the 3 more carbon will be fixed with the RUBP, and the 3 carbon containing 1, 2, 3, 4, 5 carbon containing molecule RUBP, which contain 3 molecule, will ultimately here is the 6 into 3 is 18, and due to the 3 carbon addition, and this is splitting the molecule 3 PGA phosphoglycerate, and this is the phosphoglycerate will be reduced in the form of using of the 6 ATP. Here is the stroma of the chloroplast. So the 6 ATP used to produce and 6 NADPH used to produce after the carbon fixation to stage reduction to produce 6 glyceraldehyde 3 phosphate, that is the glucose molecule. And in this way the regeneration will be occurring in this way. The regeneration in this way, here you can see this is the 1 molecule G3P will be separate and used for the production of glucose by 2 G3P, but the regeneration require 3 ATP to produce again RUBP. So this cycle will be occur. Let's begin to understand G3P will combine together to produce glucose and starch. So here is a G3P. This G3P is basically is known as the glyceraldehyde 3 phosphate, which that is 2 molecule after the 2 cycle completing of the Calvin cycle. So the 2 Calvin cycle will run to produce 2 G3P, and these 2 G3P in the presence of triose phosphate isomerase enzyme used to produce 2-dihydroxyacetone phosphate. This is the gluconeogenesis process, and in this way the aldolase enzyme used for production of 2-dihydroxyacetone phosphate to fructose 1,6 bisphosphate. So this fructose 1,6 bisphosphate contain 2 phosphate group to produce the fructose 6 phosphate. In this way the fructose 1,6 bisphosphate will be converted into the fructose 6 phosphate, and this will lead to phosphohexose isomerase enzyme used to produce isomerization to produce glucose 6 phosphate. This glucose 6 phosphate is basically the glucose 6 phosphate will produce the glucose simple because the glucose 6 phosphatase enzyme used, and in this way the directly here you can see will produce with the fructose 6 phosphate or this step. So this glucose will be converted into the sucrose, and starch synthase enzyme used to produce the starch in the amyloplast, and